Ever caught yourself wondering why your gut seems to get, well, a little more temperamental as the years go by, you know? Or maybe you thought, like, there has to be a way to soothe that internal rumble without resorting to, I don't know, drastic diets or endless supplements.
Yeah, absolutely. If that sounds like you, well, you're definitely in the right place today.
We're diving deep into some really groundbreaking work. It's coming out of the Buck Institute for Research on Aging.
Right.
They've sort of zeroed in on this key player linking the aging process to that, you know, often uncomfortable gut inflammation.
And that player is an enzyme called ADAM19.
ADAM19, okay.
Yeah, exactly. So our goal today in this deep dive is really to get a handle on what ADAM19 actually does.
Okay.
Why this discovery is, frankly, creating sort of buzz in the scientific community, and what potential this knowledge might unlock for future treatments, for age-related gut stuff.
Yeah, and we've been looking at the actual scientific papers detailing this research.
We have. And the findings are, well, they're genuinely fascinating. Really interesting stuff.
Okay, so to really grasp this, I think we first need to talk about something called senescent cells.
Right. Crucial concept here.
Yeah. So picture these as cells in your body that have, like, aged. They've gotten to the point where they just can't divide anymore.
Okay, stop dividing.
Now, your first thought might be, oh, great, no more uncontrolled growth, which, is partly true.
But, and there's always a but.
Exactly. These cells aren't just sitting there doing nothing. They're actually still quite active metabolically.
And they release this whole cocktail of molecules.
Right. And unfortunately, these molecules tend to, well, stir up trouble. We call this the senescence-associated secretory phenotype.
Or SAP for short. Much easier.
SASP, yes. And it's this SASP that's a major contributor to inflammation in the body.
Yeah, and it's becoming clearer and clearer that this kind of persistent, low-level inflammation, sometimes called inflammaging, plays a really big role in lots of age-related problems.
And that definitely includes issues we feel in our gut, right?
Absolutely. The gut seems particularly sensitive to this.
Okay, so how did researchers even, stumble upon ADAM19 in the first place? It seems kind of specific.
Yeah, it's a bit unexpected, actually. Their investigation kicked off with fruit flies.
Fruit flies? Seriously?
Seriously. Those tiny Drosophila melanogaster, they were looking at genes that influenced how much gut damage these flies got after being exposed to radiation.
Huh. Radiation damage in flies.
Well, what's fascinating here, and you see this a lot in biology, is how much we can learn about fundamental processes by studying these quote-unquote simpler organisms.
Like fruit flies.
Exactly. Many of these core mechanisms are surprisingly well conserved across different species, including us.
Right. Okay, so fruit flies and radiation. What did they find?
So within these flies, they identified a particular gene they called meltrin.
Meltrin.
Meltrin. Now, this meltrin gene turns out to be the fruit fly equivalent, the homologue of our human ADAM19 gene.
Ah, okay. So meltrin in flies is like ADAM19 in humans. Got it.
Precisely. And this is where it gets really interesting. When they figured out how to essentially turn off or deactivate meltrin in these flies,
What happened?
the results were pretty remarkable, actually. These flies, the ones without the active meltrin gene, showed a significant improvement in their intestinal barrier function.
Meaning, their gut lining was stronger, less leaky?
Exactly. Less leaky gut, basically. They also showed less inflammation in their gut tissue.
Wow.
And fewer of the molecular signs that indicate cellular senescence was happening.
Okay, so turning off the fly version of 8AM19 helped their gut stay healthier after stress.
That's the takeaway. This initial finding was a really strong indicator that this gene, and by extension ADAM19 in us, seems to play a pretty critical role in this whole aging and gut health connection.
Okay, so 8AM19, it's not just a random string of letters and numbers, right? You said it's an enzyme.
Right. The study identifies it as a metalloprotease.
Metalloprotease. Okay, for those of us whose high school science is a bit hazy, what does that mean?
It basically means it's an enzyme, a protein itself, that has a specific job,
Right.
breaking down other proteins.
Huh?
It often uses a metal ion, like zinc, to help it do its job.
Okay, like molecular scissors.
That's a great analogy, actually, yes. So the researchers started thinking, how could these molecular scissors be involved in releasing those inflammatory SASP molecules from the senescent cells?
Right. How does it connect?
Their hypothesis, their proposed idea, was that ADAM19 sits on the surface of these senescent cells,
Okay.
and it acts like very precise scissors, targeting and cutting specific proteins right there at the cell surface.
Snipping them off.
Exactly. And this cutting action is what then allows those SASP factors, those inflammatory molecules, to be released from the cell membrane into the surrounding tissue.
Ah, so ADAM19 is like the gatekeeper or maybe the releaser for these inflammatory signals.
That's the idea they were working with, that it's directly involved in letting the SASP out.
That makes a lot of sense mechanistically. Okay, so they saw hints of this in fruit flies, but, we're not fruit flies. Did this translate to mammals? Like to us?
Yeah, that was obviously the critical next step. You had to see if it holds up in more complex systems. So to explore this further, they moved to mice, and they used a drug called Batimastat.
Batimastat? Okay, what does that do?
Batimastat is known to be an inhibitor, a blocker of enzymes like ADAM19, so it stops the scissors from working, essentially.
Gotcha. So they gave mice Batimastat. Then what?
Then they exposed these mice to chemotherapy.
Ah, okay, which is another kind of cellular stress, right? Like the radiation in the flies. It can also cause cells to become senescent.
Exactly. It's a well-established model for inducing senescence and related damage, including in the gut.
So what were the findings in the mice? Did blocking ADAM19 with Batimastat actually make a difference after the chemo?
It absolutely did. It was quite clear. The mice that received the Batimastat, the ADAM19 inhibitor, showed, again, stronger gut barriers.
Less leaky gut again.
Less leaky gut. And significantly lower levels of inflammatory markers in their gut tissue compared to the control mice that didn't get the drug.
Wow. So this is where it starts to look like maybe a potential treatment strategy.
Exactly. This is where the therapeutic angle really starts to take shape. Because here you have a drug targeting ADAM19, and it has a clear, measurable, positive effect on gut health in a mammal model after a significant stressor.
That's a really significant step, moving from flies to mice and seeing the same kind of protective effect. But I remember the research didn't stop there, did it? They also looked at human cells.
Correct. They took it to the next level.
Yeah.
Human cell cultures in the lab.
Same story.
Pretty much, yes.
Right.
When they inhibited ADAM19 activity in cultures of human cells, they observed very similar results to what they saw in the flies and mice.
Like what specifically?
They saw a reduction in those telltale signs of cellular aging. That included lower levels of those harmful SASP proteins we've been talking about.
Okay.
And also a decrease in the activity of something called β-galactosidase.
Beta-galactosidase. I've heard of that. That's like a classic marker for senescent cells, isn't it?
It is. It's widely used. So seeing less of it active suggests that inhibiting ADAM19 was somehow reducing the senescent characteristics of the cells.
So, okay, we're seeing this consistent pattern. Flies, mice, human cells in a dish. Blocking ADAM19 seems to lessen senescence markers and inflammation.
Right. It's building a strong case.
Now, the study uses this term, senomorphic strategy. They contrast it with senolytic. Can you sort of unpack that for us? What's the difference and why might it matter?
Yeah, that's a really important distinction they make. So senolytic therapy is analytic, meaning to destroy or break down work by directly trying to kill and eliminate senescent cells from the body.
Get rid of them entirely.
Get rid of them. The idea is remove the source of the problem. Now, this new approach targeting ADAM19 is different. They call it senomorphic, meaning relating to form or structure, or in this case, behavior.
So changing their behavior, not killing them.
Exactly. The goal isn't to destroy the aging cells, but to change how they behave, to make them less harmful, specifically by reducing their release of those damaging SASP substances, those inflammatory molecules.
Ah, okay. So you leave the cell there, but you stop it from secreting the bad stuff.
Precisely. And this offers potentially a more nuanced approach, maybe even a gentler one.
Why gentler? What's the potential downside of just killing senescent cells?
Well, we're still learning a lot about senescent cells. They might actually have some beneficial roles, at least temporarily, like in wound healing or tissue remodeling, just wiping them all out.
Might have unintended consequences.
Could do. The long-term effects aren't fully understood. So the senomorphic idea is instead of this sort of scorched earth policy of eliminating the cells, let's just modify their harmful output. Let's manage them.
That makes a lot of sense. It's like reining them in rather than just wiping them out entirely. Okay, so getting back to the mechanism. The study dug deeper into how ADAM19 actually helps release these inflammatory signals, right?
Yes, they wanted to confirm that molecular scissors idea.
And did they?
They did. They provided evidence pinpointing that ADAM19 directly helps release specific SASP proteins. It does this by essentially cleaving them, cutting them right at the cell surface.
So it is the scissors.
It appears to be acting precisely like that.
Yeah.
This is the direct mechanism they were looking for, showing how this enzyme actively contributes to the inflammatory environment these senescent cells create.
Molecular scissors for inflammation. I like that visual. It really sticks.
Yeah, it captures the action quite well. It's snipping those pro-inflammatory proteins loose from the aging cells.
And did they figure out which proteins it was cutting? Was it just snipping everything, or was it specific?
Very specific, it seems. They use a technique called proteomic analysis, basically, studying all the proteins present.
Okay.
And through that, they identified a specific set of 12 SAMP proteins whose levels were significantly reduced when ADAM19 activity was blocked.
12 specific ones? That's quite targeted.
It is. And it's a key finding because it really lets us narrow down which molecules ADAM19 is unleashing that are likely causing the trouble downstream, contributing to the inflammation.
So what's the significance of these particular 12 proteins? Are they just random inflammatory molecules, or are they known players in gut problems?
That's the really compelling part. Many of these 12 proteins they identified are not unknown.
Ah.
They've already been linked quite strongly to inflammation, immune responses, and even tissue remodeling processes in diseases that affect the gut,
Like what?
like inflammatory bowel disease, IBD, and Crohn's disease.
Wow. Okay, so these aren't just vague inflammation markers. They're proteins implicated in specific, serious human gut diseases.
Exactly. And that connection really highlights the potential real-world relevance of this research. It's not just about the general aches and pains of aging gut. It suggests that targeting ADAM19 might be relevant for people living with chronic defined gut conditions too.
That could be huge for those patients.
Potentially, yes. It suggests a new therapeutic target that might apply not only to age-related decline, but also to these established inflammatory diseases.
Okay, so let's step back a bit and look at the bigger picture here. What's the potential impact of all this research on how we understand aging and gut health?
I think the potential is pretty substantial. It genuinely offers a new way, a new strategy for thinking about how we protect gut health as we age, and how we might reduce that inflammation driven by aging cells, but without resorting to just killing those cells off.
The senomorphic approach.
The senomorphic approach. And this could, down the line, pave the way for developing novel treatments, not just for the gut issues that crop up with natural aging,
Right.
but also potentially for the kind of gut damage that can happen as a side effect of things like chemotherapy or radiation therapy, which we know can induce senescence.
So it could help protect the gut during cancer treatment, for example.
That's certainly one potential application worth exploring based on these findings.
So yeah, going back to the initial question you posed, can we calm gut turmoil without drastic measures? This research seems to be pointing towards exactly that kind of possibility.
Okay, so let's just quickly recap the main points for everyone listening.
Good idea.
Researchers, mainly at the Buck Institute, have pinpointed this enzyme, ADAM19, as a critical player in gut inflammation linked to aging.
Right. It acts like molecular scissors, helping senescent or aging cells release harmful inflammatory substances, the SASP.
And crucially, experiments showed that inhibiting ADAM19, blocking its action, led to reduced inflammation and healthier gut barriers.
Yeah, and they saw this consistently in fruit flies, in mice, and even in human cells in the lab.
And we talk about the senomorphic nature of this strategy.
Which is key. It's about changing the behavior of these aging cells, making them less harmful, rather than just eliminating them outright like senolytics aim to do.
And that might be an advantage, potentially safer or more nuanced. It could be, yes. It's a different philosophical approach to tackling age-related cellular changes.
It's truly fascinating stuff. It really makes you think, doesn't it, about the possibility of influencing aging effects at this really fundamental cellular level. But without needing super aggressive interventions. And it opens up questions like, what other age-related processes might be governed by similar kinds of regulatory mechanisms? Are there other scissors we don't know about yet?
Absolutely. It raises important questions for future research, definitely.
Yeah.
And maybe just for how you personally think about the intricate dance between aging and health.
A lot to chew on there. Well, thanks for taking this deep dive with us today into the world of ADAM19 and gut health.
My pleasure. Always fascinating to unpack these studies.